Hi, welcome back to Protein and Enzyme Function in Biochemistry. My name is Kevin Tokoph. Make sure to like this video and subscribe to the channel for future videos and notifications. Alright, so in this video we're going to talk about antibodies. And we're not going to really go into so much super detail on the immune system, how they're made, how they're modified, created, and so forth. We just want a very general understanding because in biochemistry we don't just talk about them necessarily in the context of the immune system within a living system. We also extend that into the lab because there's also unique things that we can do with antibodies in the lab. So I have here a very general sort of a cartoon structure here of an antibody. And we can sort of talk about the various parts of an antibody because they're all really important. All right, so antibodies in general, when you just look at a single one, they kind of look like a Y. So all these little scribblies right here, these purple scribblies, these two up here, and then the blue squiggly down here, that's the actual antibody. This pink circle right here, this is not part of the antibody. We'll talk about that in a minute. That's actually called the antigen. Okay, so what is an antibody? An antibody is a protein. They're actually glycoproteins, if you note these carbohydrates right here. Antibodies are glycoproteins that play a role in the immune system in most upper-level organisms. That includes humans. And antibodies, what their function is they're made by certain cells. Some of them are called B cells, or plasma cells, I should say, which are activated. They're immune cells. And these antibodies bind proteins. Now, they don't just bind any proteins. Generally, what they bind in normal homeostatic conditions is they bind foreign proteins. So it could be a protein from a foreign bacteria, it could be a protein from a virus, or something that's not really talked about a lot. There are actually many people that are allergic to dairy. There's in cheese, milk, and so forth, there's a protein called casein. You may have heard of it. A lot of people, including myself, have antibodies that bind casein. In people even that don't have celiac disease, there are many people who have antibodies that bind gluten, which is also a protein. Okay, so antibodies bind foreign proteins, and certainly from a dietary perspective, anything you're eating from the diet is considered foreign. Okay, now how do antibodies bind? Number one, they have these regions up here, which are called variable regions. Okay, these are actually, sometimes they're called hypervariable regions specifically, and these are the parts right here that have the capacity to bind the antigen. Now, what is an antigen? An antigen, notice it's not an antibody. The antigen is the substance that the antibody binds. Antigens are generally proteins or small fragments of proteins, usually about 8 amino acid residues long. They're not very large parts, but these antigens have specific amino acid sequences. This pink thing has a certain amino acid sequence that's complementary to the variable region of the antibody right here. Okay, it's complementary, and so the antigen sticks on the antibody. And one thing that's really important is the antibody actually binds the antigen very tightly. It's very tight binding, and that's what allows antibodies to be so effective at binding these antigens, and then from there tends to be an immune response. Okay. We actually have a video somewhere on this channel about the complement system, which is an immune system response to attack antigens. And you can watch that, but suffice to say antibodies can activate the complement system, but only when they're bound to antigen. And in fact, the antigen binding to the antibody is a signal to the immune system that hey, we have a foreign invader, and we need to neutralize it and get rid of it, basically. All right, so just a little more details. These purple regions, these arms of the antibody, things that make up the top of the Y, are called the FAB. And that means F antigen binding, or fragment of antigen binding. It turns out you can actually fragment these regions away from each other, and these fragments become antigen binding fragments. And that actually makes sense because on the variable regions of the F sub AB, this is where the antigens bind. Okay? This region down here is called the constant region. This C is actually not for constant. It's a misconception. It's actually for crystallization. That's not important. But the C can help you remember constant. This region is constant. A lot of these regions, particularly up here, change depending on the antibody. In other words, they change depending on which antigen it's designed to bind. But this region down here is always constant. Okay? And there are also some other constant regions elsewhere which we won't go into. Okay, but the thing that you generally need to know is that antigens bind to these variable regions up here. The other interesting thing about antibodies is that they have carbohydrate residues on the constant region. Down here, they have carbohydrate residues. The carbohydrate residues, their exact, or I should really say their range of functions are not known, but one very important function is known, is that the carbohydrates play a role in complement system activation, specifically of the protein C1, which is a complement protein. Okay? So this is the general structure that you need to know. This is the FC region, these are the FAB regions, these are the variable regions up here, and this is where the antigen binds. Okay? Now we're going to do something a little bit different and go to a PowerPoint presentation. So the key here is that antibodies tag and trap antigens. What does it mean to trap them? Well, number one, the antibodies bind antigens, and they do so very, very tightly and specifically. One important thing here is that if you have an antibody that's specific for, say, a bovine casein protein, that antibody will only bind bovine casein protein. Okay? If I have an antibody that's specific for, I don't know, some weird enzyme, then that antibody will only bind that weird enzyme. Okay? And usually it's going to be a foreign enzyme because antibodies tend to only bind foreign proteins, not our own proteins. And we say they tag these antigens because, in general, the reason you want to tag them is so certain immune cells, such as macrophages, know to destroy those antigens because they're often linked to foreign invasions by bacteria and viruses and so forth. Okay. Now, if I go back real quick, and if I look at this region on the antigen right here, the region on the antigen, the amino acid sequence, in other words, to which the antibody binds, this is called the epitope. Okay, epitope. Another word or set of words you'll hear this referred to as antigenic determinant. All that refers to is the amino acid sequence of the antigen to which the antibody binds. And this antibody, this variable region of the antibody, will only bind this particular epitope, which means it only binds that antigen. Okay, and that's part of the reason it's so specific. All right, we're only going to go into two of these antibodies here. The most important ones are IgM and IgG. Now, IgM is going to exist as a pentamer of antibodies. Okay, there's going to be 5 of them together. And the thing about IgM is they're released very early in the immune system response. Okay, they don't have as strong binding as IgG, and in general, IgM seem to be more a part of the primary immune response when you're first exposed to this particular antigen for the first time. And so IgM, they are made very quickly to combat that particular antigen. If it's a bacteria, you have a bacterial infection. IgM is made rather quickly, and also you could also kind of think of it as a haphazard synthesis with respect to the others because it's made so fast to try to combat that initial exposure to the antigen. The IgMs are not as specific. The IgMs are not as tightly bound to antigens. They don't have the capacity to bind as tightly. They can, though, activate the complement system, as it says here. IgG exist as monomers, and they're part of the secondary immune response. And so that means that when IgM is initially made to combat the initial exposure to an antigen, you have these cells which are called memory cells. That's not that important. But memory cells, they remember that antigen. And they make IgG, which is a monomer, in preparation in case that antigen comes into the system again. So in other words, let's say you get a strain of influenza that you've never seen before. Well, if your body has never seen that before, it's initially going to make IgM to combat it. But in the process of doing this, which would be the primary immune response, your memory cells, which are going to be generated, are going to remember that antigen and start making IgG against the same influenza antigen. So if you're ever exposed to that strain again, you likely won't get sick because you have this IgG. And the thing about IgG is it binds much more strongly than IgM. And you tend to make a lot of these. So that's the reason why you generally don't, for a lot of things, don't get sick a second time. It's because you have memory to it, and the memory is carried in this IgG antibodies. Okay? There are also some others here. We're not going to focus on them so much, but IgM and IgG seem to be the most important, and IgG also, like IgM, can activate the complement system. All right? And this is something we already went over in the paintbrush picture, but these right here, this is a space fill model. This is a ribbon diagram on the left, but you can see in the space fill that these are the antigen binding regions, and this is the Fc region of the antibody. Okay. Also, something I mentioned, the Fc region has carbohydrates on it, and the purpose of those seems more to be in terms of protein recognition, as in the complement system proteins like C1, so that those complement proteins can be activated. It's actually known if you remove the carbohydrates, the complement proteins don't necessarily get activated to anywhere near the same extent. And then specifically here on the FAB regions, you have the antigen binding sites, and these are the spots to which those pink circles I showed you, those antigens actually bind. One thing to keep in mind is that remember that these antigen binding sites for these antibodies are absolutely specific for one particular antigen. So if I have antigen 1, antigen 2, antigen 3, only antibody 1 will bind antigen 1. Only antibody 2 will bind antigen 2. Only antibody 3 will bind antigen 3. Antibody 1 will not bind antigen 3, if that makes sense. So every antigen is going to have an antibody that binds it. And they're very, very specific and bind very, very tightly. This is another representation of this. These up here, these are the antigen binding fragments up here, and then this is the crystallization or Fc fragment. Okay, you can see the Fc fragment has carbohydrates on it. Notice that each of these, if I actually look at this, these chains in white here, these are called the light chains. And these longer chains right here, they're actually connected by a hinge region. These are called the heavy chains. Okay? In general, the constant regions, this is why it gets a little confusing. This is the Fc region, and the C stands for crystallization. However, constant is, in general, it cuts off like right here. So if I also draw a line right through there and then right through there, everything in this region where you have these C's right here, that's the constant regions. Okay, the V on the edges here, that's the variable region. And these are the only regions that actually tend to change depending on the antibody. And this is also where the antigen binding site is, note. So the C here is where the constant region is, the V is for variable. Okay, but it just turns out that these two right here, that's the Fc, but the C is not constant, it stands for crystallization. Okay, so it's a little confusing. And then these two parts right here, you can see are connected by a hinge region. It also turns out that there are some utilities, and if you ever want to cleave this hinge region. In fact, there are enzymes that you can use to cleave it, and it allows you to individually study the Fc fragment or the FAB fragments, and sometimes that's very useful. Okay? So hopefully that makes sense. And one thing that's really important here is that when antibodies bind to an antigen, it's not covalent. It's all done through your weak interactions, okay? So this is a good representation right here. This right here is the antibody, and this is the antigen, okay? The orange thing right here is the antigen. We're just going to assume that. So basically right here on the antibody variable region, that is, notice we're going to have a nonpolar region. Maybe here we have a full positive charge. Then we have hydrogen bond donors over here. Well, remember, if I expect the antigen to bind this tightly to the antibody, it ought to be complementary. Well, what's complementary to a nonpolar amino acid? Well, other nonpolar amino acids, because they form van der Waals interactions. If I have a full positive charge here, maybe I want a full negative charge over here. That's a Coulombic interaction or a charge-to-charge interaction. Over here, if I have a hydrogen bond donor amino acid, over here I want a hydrogen bond acceptor amino acid, so hydrogen bonding. So we can have full electrostatic interactions, hydrogen bonding, and van der Waals interactions. And in general, the antibody is synthesized specifically to bind to the particular antigen. Okay? And turns out there are ways that are not yet understood that this region of here on the antibody, these amino acids can actually be tweaked. And that the sequence changes to alter the specificity for particular antigens. Okay? And the only thing else I'm going to do in this is just hopefully give you more of a space fill picture of this. This over here, these actually right here, these are parts of the antibody. This thing that's sort of red, greenish over here, okay, this is the actual antigen. And just hopefully what you can see here is that the variable regions right here, these two variable regions, they're going to hold on to this antigen, okay? You can kind of see them almost wrapping around there. It gets in there pretty tightly, okay? And the basis of it ultimately are your amino acid residues. So this is an asparagine right here. Asparagine is going to be a hydrogen bond donor. And then over here you'd have a hydrogen bond acceptor. Arginine here is going to have a positive charge, so likely here you have a negative charge on the antigen. And so you have amino acids interacting with amino acids of the antigen, and ultimately this provides very, very tight binding of the antibody to the antigen. Okay? So hopefully this gives you a really good look at the principles of how antibodies bind to antigens. Okay? And in the next few videos, we're going to look at some applications of antibodies for biochemistry. That's going to include ELISA and Western blotting. And if I can think of any other important ones, we may do those as well. Okay. And we'll have a completely separate playlist for actual antibodies in their relation to the immune system in a living system. So hopefully this video helped. Make sure to like this video, subscribe to the channel for future videos and notifications.